So, hello everyone, welcome back to the final session of the day. I always find this is often the most difficult session for everybody to stay awake for, although we were just commenting with the speakers that no one's got the excuse of trains to catch or flights to catch, no one's got to check out of a hotel. So I'm hoping that despite the fact we're running a little bit behind, you're all going to be able to stay and listen. We've got two excellent speakers this afternoon, and they are talking on the area on which I always am really interested at these meetings. I see patients and diagnose them with mesothelioma on a not infrequent basis. And one of the first questions is always, what treatments are there? What can you do for me? And so I'm always interested to hear of the new research coming up. As you probably know, we've not had many good news stories from clinical trials in recent years, but I think Professor Dean Fennell is going to talk to us about some really exciting results that we've had, and hopefully one of the newer therapies for mesothelioma coming on board soon. So I'm really interested in his talk today. Before I hand over to Dean, I would like to just thank our sponsors for this session, which is Digby Brown, and they've enabled us to have all of the AV support for this meeting, which has been fantastic from a speaker perspective particularly. So thank you very much to our sponsors. And I'm going to hand over now to Dean. Professor Dean Fennell is a professor in thoracic medical oncology, and he's director of the Leicester Mesothelioma Research Programme. And really what he doesn't know about mesothelioma clinical trials and research is probably not worth knowing. So I'm sure he's going to give us a very excellent talk this afternoon. Dean.
Am I unmuted? Is that okay? Good. You can hear me. Okay, great. Well, again, thanks very much indeed for the organisers for giving me this opportunity to speak. This is a special year for mesothelioma in terms of clinical trials. I'll try and explain why in the next few minutes. So what I'll do, actually, I'll divide the talk into these three sort of treatment settings. First encounter in terms of drug-based therapy for mesothelioma, the frontline setting, what we do to try and sustain control of the cancer actually following chemotherapy with maintenance, as there's new data there, and the relapsed mesothelioma setting, where there are developments afoot which are looking very encouraging. I'm going to start then in the frontline setting with this historic data. This is a summary of the clinical trials. I want to focus a lot on phase 3. We need phase 3 trials in order to change practice. These are large, often randomized studies in which we have a control arm which we're comparing our drugs to. And you can see here these 4 studies. The first is the definitive trial of chemotherapy. This was cisplatin pemetrexed versus cisplatin alone. And I want to show you this hazard ratio. This is 0.77. So this is a measure of the difference between these curves. If it's 1, they're completely overlapping. And as you approach to 0, these curves separate with a doubling of survival time if this is around 0.5. So as you can see here for this combination, a very similar combination, antifolate 0.76, and then the addition of this drug bevacizumab. This was never actually licensed. This is the MAPS-1 study from our French colleagues. And then this is really what I'm going to focus on here today. This is a historic study, FDA approved in only the last few weeks. And this is a combination of immunotherapy with an anti-CTLA-4 drug, ipilimumab, and nivolumab. This is a bold trial comparing combination immunotherapy against chemotherapy. And you can see actually numerically the hazard ratio 0.74 is better than all previous studies by a small margin, but nevertheless superior overall for this difference. So I'll just give a little bit of background around the sort of potted history actually of the immunotherapy of mesothelioma. There have been studies that have focused really on the relapse setting with monotherapy. Many of you will be aware that pembrolizumab, Keynote 28, was perhaps the first and most prominent early study of immunotherapy. And then we have a number of other trials that I'll just point out. Lower down here, you'll see the phase 3, which I want to emphasize. The first actually was an anti-CTLA-4 trial. This was using tremelimumab versus placebo, and that was robustly negative. suggesting that at least in the relapse setting, single agent anti-CTLA-4 does not seem to have any activity. Similarly, actually, when looking at PD-L1 or PD-1 inhibition, this is the recent PROMISE study. Now, this wasn't against placebo as determined. This was actually against chemotherapy with either gemcitabine or vinorelbine. But essentially, immunotherapy versus chemotherapy showed no difference for PD-L1. So not the most exciting, two negative studies really for single agents. Here in PROMISE, I think the control arm was the factor. Here, really, we didn't see any definitive activity for anti-CTLA-4. However, based on trials in phase 2 settings, single arm studies, or in this case with MAPS2, randomization of combined immunotherapy, combining these two classes, ipilimumab, nivolumab, and comparing that to nivolumab alone, there was a clear separation of the progression-free survival overall survival curve, suggesting that there was indeed synergy with those two drugs. So these two trials, INITIATE and MAPS2, really underpinned the Checkmate 743 study. And here we can see some of the data actually around survival. I'll just draw your attention here to the overall survival. INITIATE, they didn't even measure it. Patients going out to, I can't remember how many months, probably 20 odd months or so, didn't reach the median survival. MAPS2, we see a 15.9 month survival in the relapse setting, this is. And with Checkmate 743, we see 18.1 months. But I'll show you now sort of the key data. Before I do that, though, the response rate you can see in Checkmate 743, 40%, very similar to what was originally published back in 2003. And a respectable disease control. This is a combination of shrinkage and stabilization of the tumor at 76.6%. Now, this is the core result to draw your attention to. On the left here is the survival for patients with non-epithelioid mesothelioma. And you can see here a big difference between the curves, big space between the curves. And the hazard ratio for this was very, very impressive, actually, at 0.43, I believe, compared to the sort of epithelioid sort of subgroup, which of course is the majority of patients, where the hazard ratio was actually slightly, well, it was 1, or sort of close to 1. Now, what does this tell us? Well, it tells us actually that the immunotherapy is working approximately the same in the non-epithelioid and the epithelioid subgroups. If you see in this table here, 18.1 months overall survival for non-epithelioid, 18.7 for epithelioid, and the hazard ratio here is 0.93. So what we're saying is that this brown curve here and this brown curve here are indistinguishable. However, when we look at chemotherapy, we can see that in the non-epithelioid group, the survival is pretty appalling, actually, at 8.8 months. I think it's something we're all aware of when we treat patients with chemotherapy with non-epithelioid. It's not the best treatment for controlling this. But with the immunotherapy, we see 18.1. And when we compare the non-epithelioid to the epithelioid, chemotherapy is working so much better. And this hazard ratio, based on the histological differences, is 0.47. So this histology appears to be negatively prognostic. It's basically associated with a poor outcome in the control arm, whereas the experimental arm is doing okay, but it's not doing better because of non-epithelioid histology. Now, just showing you here actually the tail of the curve. This is encouraging. Many patients, you can see as circles here, haven't progressed yet, haven't died yet. And that probably means actually that as we go over time, this was an early read of data. It was a premature, immature data in the sense that the data was positive before time. But some of these patients who haven't progressed, if we wait perhaps for another year, we may see this curve move away, possibly even further from the control arm. So this is going to be very interesting. You can see here going out to 36 months, 3 years in non-epithelioid patients, a plateau of around about, well, almost 30%. These patients appear to be long-term survivors, certainly for non-epithelioid mesothelioma. It's the first time we've seen this in history, I think. So what about safety? Well, there's really, in summary, no major difference actually between the two groups. I have a serious treatment-related or treatment-emergent AEs, and we can see here 5% or less, slightly less for chemotherapy, but 5% for ipilimumab and nivolumab. So in general, a well-tolerated treatment. So I think if looking at the risk-benefit, we can see here in non-epithelioid, well, there's no question these patients are doing so much better with ipilimumab and nivolumab. In the epithelioid, there's a bit more equipoise, really. No significant difference in overall survival between chemotherapy and ipilimumab and nivolumab. And possibly just the faintest hint of a little bit more in the way of toxicity in that subgroup. I think it's really important to mention that this is an intention to treat analysis. The FDA gave their approval because the overall hazard ratio was 0.74, and that was for the total population. There wasn't a pre-planned stratification in the sense of separating out a group that was powered for better survival, let's say. So that's really why I think the FDA gave the approval, but I think the signal, as you can see, comes clearly from the non-epithelial mesothelioma subgroup. So what is driving this chemo resistance? Well, we've known for some time actually that sarcomatoid biphasic mesotheliomas do badly in general. This is with or without chemotherapy. They're more invasive, highly proliferative, and of course drug resistant. And one of the phenotypes that we've seen over the years with literature is that these non-epithelioid mesotheliomas exhibit features of what is known as epithelioid mesenchymal transition. This is a feature, very often stem-like feature, associated with all those characteristics that I described in sarcomatoid mesothelioma, particularly this invasive, highly proliferative, drug-resistant phenotype. And it is likely that with enrichment of EMT, as has been shown by Didier Jean in France and his colleagues using sequencing, gene expression analysis, this enrichment of the EMT signature in the non-epithelioid mesothelioma is a prime candidate for underpinning the drug resistance, I think, that we see in the Checkmate 743 trial. PD-L1, still very unclear, actually. I think the feeling from Checkmate 743 is slightly prognostic. And, you know, the trial initiates, it was clearly with 22C3 PD-L1 predictive from this single-arm study. Not so clear from the MAPS-2 study. And if we look at the data from the Checkmate 743, we can see below here, PD-L1, less than 1%. Patients are doing a little bit better, going towards this line here. And patients with PD-L1 are doing a little bit better. And the reason they're doing better is predominantly because the survival in the chemotherapy arm is worse. You can see 13.3 months here compared to PD-L1 less than 1%, which is 16.5 months. So doing slightly worse if they have PD-L1. And we can see that the high PD-L1 maybe is a bit predictive. Maybe the survival is just a little bit better than low PD-L1, 18 months versus 17.3, but there's very little in it. So I would say that PD-L1 is acting here as a negative prognostic factor, not as a positive predictive factor as we see in lung cancer. So what next? Well, we're seeing a number of studies currently exploring chemoimmunotherapy. Perhaps the most prominent of those is the DREAM trial. This is actually looking at a schedule which has been successful in its scheme in lung cancer, which is combining standard chemotherapy with immunotherapy. Now, we've not seen a randomized trial yet, but two trials, the DREAM trial and the US PRECOG study, both have examined this combination. And indeed, the Australian team led by Anna Nowak published in Lancet Oncology only very recently on these results. We have the IN227 study from Canada, which is pembrolizumab combined with pemetrexed cisplatin. Now, this is a randomized trial compared with chemotherapy alone. And I would imagine we'll get a readout in 2021 for this, but a very important phase 3 trial. that is similar in design to something called the Keynote 189 study, which led to an approval in non-small cell lung cancer in the frontline setting. Other trials, the Beat Meso study is of course being run at the moment in the UK. This is an important study because it's based on a very similar scheme as we saw in the Empower 150 trial in lung cancer, which again was positive and changed practice. Here we're looking at bevacizumab and chemotherapy, the MAPS-1 combination, combined with atezolizumab immunotherapy in the experimental arm, compared with this MAPS-1 triplet. This may well be a positive study. I think one of the questions we'll have to explore at a later date is whether or not, because this control arm is not licensed within the UK, what the cost effectiveness of that combination is going to be. But nevertheless, this quadruplet treatment could push survival out significantly. And I have to say, it's going to be the survival that ultimately dictates, I guess, where this treatment goes. But very exciting study. We've got a study. We completed it actually in February. It's an important study. There have never been any treatments which have improved survival in the relapse setting in mesothelioma. So we've looked at nivolumab, which actually happens to be licensed in Japan as a monotherapy. It's the only country in the world where that's licensed. The NHS, of course, is offering this currently until March of next year. But we've looked at this in a 2-to-1 randomized study against placebo, and we're presenting the data for this in ILSLC in January. What's next? How do you advance on Checkmate 743? It's a fantastic result. Well, we have to probably look to lung cancer to see what's down the road. I think this is almost in the post, actually. I can't imagine this not being another design that's coming soon. This is where chemotherapy, only 2 cycles, was given with ipilimumab, nivolumab, and showed a significant improvement over chemotherapy alone. Now, this combination actually has been FDA approved, and the chemotherapy is working as an immunomodulator. The DNA damage is triggering an inflammatory response through something called the STING-gas pathway, and that almost certainly seems to be priming the tumor actually to respond better to ipilimumab and nivolumab. And so this could well be something we see in mesothelioma in the near future. There is another study I want to draw attention to briefly, which is the ATOMIC trial. It takes a completely different approach, actually, and it's one I've spoken about a number of times before, and I'm sure you've heard about. This is a trial that is actually exploiting a vulnerability in mesothelioma due to a loss of an enzyme. And this vulnerability can be exploited using a drug called arginine deaminase. This is data from patients in the ADAM trial where there was a clear improvement in progression-free survival and indeed overall survival when patients received the arginine deaminase alone. These patients were selected on the basis of this biomarker. And this biomarker is predominantly in non-epithelioid mesothelioma. So by selecting these patients on histology, this phase 3, a global phase 3, that's really emerged from Peter Slotarek's work in London, will hopefully provide some evidence as to whether or not this can improve benefit of chemotherapy. So another approach for targeting non-epithelial. So let me very briefly move on to maintenance. There's been very little, active drug therapy for maintenance developed over the years. These are the slew of negative trials. We've got the flutamide, pemetrexed data here, FAK inhibitor study, COMMAND, which are all robustly negative. Just to remind you, the purpose of maintenance is to really try and control the cancer beyond chemotherapy. We normally give no more than 6 cycles of chemotherapy, and then it's really up to the biology to determine when that tumor comes back, because no drug is being given to suppress it. And having something after chemotherapy would be a good idea. Well, this fabulous study from the Dutch group, Navalt 19, looked at gemcitabine monotherapy randomized against best supportive care. And you can see here, I think, an incredible hazard ratio of 0.43 in favor of the gemcitabine. This was a phase 2 trial, not a phase 3, but I have a feeling based on these statistics, actually, and this distance between curves, that if this were expanded now into a larger study, we would see a positive phase 3. This also suggests, actually, that some form of active therapy the right active therapy after chemotherapy has the ability to more than double progression-free survival. We have a study where we're looking at dendritic cells. It's essentially an immunotherapy, but it's a vaccine-based therapy. This is now recruiting, and there's a trial in which we're taking cells out of the body. These are monocytes, which are differentiated into immature dendritic cells. These are then exposed to mesothelioma proteins, recombinant proteins, which then really program these cells to go back, when you put them back into the body, to seek out mesothelioma and orchestrate an immune response leading to inflammation. Based on some very promising early phase data, this is now a pivotal study that's taking place in these 5 countries, and this is funded by the EU Horizon 2020. So finally, to relapse mesothelioma, this has been the biggest challenge, really, I think, to date, because we don't have a licensed treatment. These are unfortunately trials which have been very well designed, I think, but all negative. Here, determined against placebo, vantage against placebo, practically overlapping curves. So what have we got? Well, as I mentioned, the confirmed trial is going to read out, hopefully, for January. And here we've got this 2-to-1 randomized trial against placebo. I think the goal here is to see whether or not, of course, we can improve overall survival and progression-free survival. But one of the goals here also is to try and fully delineate what is driving response in patients. And as part of the study, actually, we do have an extensive pharmacogenomic analysis. And this is something that we hope we'll be able to try and accelerate and present data on soon after the final data is produced. There's no question that nivolumab as a single agent has activity, but the question here we're trying to address is that activity and its magnitude significantly better than nothing, which is the placebo? This is just illustrating the type of responses you can get with immunotherapy. Unfortunately, not in everybody. This is what we want to try and understand. This was a patient who had heavy thorax full of disease, and you can see here over 3 successive scans, pretty much clearing of the cancer. Another study that we're doing, which isn't the most perhaps exciting based on what else is out there, but can't be underestimated, is a randomized study of vinorelbine. Again, this result should be available very shortly. And what we're looking at here is the randomization, again, 2-to-1, vinorelbine versus active symptom control. We're looking at overall survival and progression-free survival. And here we are gaining additional data really around the potential biomarker, which we think will enrich for patients likely to benefit from vinorelbine. So I won't go into detail about that, but this will be available data, hopefully published in 2021, if not before. And finally, there's a trial which we opened, over a year ago now, about a year ago, called the Mesothelioma Stratified Therapy Study. There's no doubt that mesotheliomas are highly heterogeneous, and we need ways of partitioning mesothelioma into groups that are likely to benefit from treatment. So we've started this. It's a rather crude sort of design based on the genetics, but this is a first attempt, I think, to see if we can pre-select patients based on their biology and then treat them accordingly. We've completed 2 of these studies. This is the rucaparib PARP study, which we presented at ASCO, and abemaciclib, which is a p16-linked4A targeted drug. And both of these trials, I think the rucaparib is in press. We should have the abemaciclib data to present next year. And these other trials, these are looking at novel immunotherapy combinations, but here the goal actually is to try and again use the clinical trials as a platform for understanding the genetics that underpin sensitivity to immunotherapy and all combinations. So with the PARP study, actually, we have reported this. This is a positive trial, it met its primary endpoint. There were responders, and as in order to try and understand what's driving this response, we've been using parallel models. So we're using explants. These are live bits of tissue which come from patients acquired at surgery. We can treat them with a drug like rucaparib. We can actually see activation of DNA repair mechanisms or cell death, and we can look at the heterogeneity, measuring quantitatively how they respond. And you can see non-responders here versus responders, where we can use gamma H2AX as marker to give us a signal of activity. What you can see is very satisfying because it actually shows big variation across the cohort of patients, and that's what we see in the clinic. So by sequencing these patients' tumors, we may be able to build a model of what's underpinning response. Here's an example. A patient here who had a response, you can see some dose response gradient, whereas this is completely flat, had a clonal BRCA2 mutation. So this was a ubiquitous mutation in the DNA repair gene BRCA2. And also this patient's tumor had something called signature 3, which is a feature of the genetic landscape consistent with a tumor that would be sensitive to PARP inhibitors. So this is the sort of thing we're trying to explore, but looking a little bit deeper across a broader range of tumors to see what mechanisms could be causing sensitivity. And this is finally just an example of a patient's response to rucaparib. Some patients stayed on treatment, actually, a fair proportion of the cohort over a year, having had relapse with prior treatments. And we have actually tried to rebiopsy areas of progression in order to be able to characterize differences in their genomic profile between that baseline and when this tumor has somehow escaped the effects of the drug, as long ago, as I say, as a year later. So you can just see some shrinkage here of a tumor in one of the patients. So in summary, as I say, I'll repeat again, it's been a really amazing year actually for mesothelioma. Combination immunotherapy is now the standard of care. Officially, FDA has approved that. We won't hear from NICE until August of next year what the UK decision is going to be, but I would put money on it that non-epithelioids at least will benefit from this, if not everybody. The relapse setting is still a big challenge, actually. It might become a bigger challenge if 9LA type chemoimmunotherapy designs happen, because all the chemotherapy will be swept into the front line. But this is an opportunity, actually, for researchers, I think, to try and develop studies that can improve outcomes, but we're still lacking any positive trials. And finally, molecular stratification. Well, we've seen it work in other cancers. Mesothelioma, we're still trying to learn more about these mutations and how we can target them. But we know it's feasible. We know, trials like MIST and others that I'm sure are coming will allow us to hopefully get to more targeted, more effective treatments. And we heard earlier about Nick May with a PTCH driver mutation. I mean, that's possibly what's coming down the road if we can, identify these effective therapies. So with that, I'd like to thank you all for listening, and happy to take questions later. Thank you.
Dean, thank you very much. It's an excellent talk, and it does seem like we potentially have some new exciting treatments on the horizon for our patients. So there's been a few excellent questions coming through on the chat, but I'm going to save those until the Q&A session at the end. So please do keep those coming, and it saves me from thinking of them during Kevin's talk. So to move swiftly on, I'm going to hand over to Professor Kevin Blythe, who probably needs very little introduction to those who are working in Scottish mesothelioma. So Kevin has really been a pioneer for improving standard of care for patients with mesothelioma in Scotland, and has really worked phenomenally hard to get the Scottish Mesothelioma Network up and running. And I know it's been probably a labour of love over many years. But it seems like now he's got a new labour of love that he has to concentrate on for the next 5 years. So I'm going to hand over to Kevin, who's going to talk to us about Predict Meso, his international accelerator network.
Thanks very much, Beth. And hi, everyone, again. So I'm kind of quite excited to tell you about the accelerator. So it's been a program that we've been working up now for probably 2 years, and was funded by Cancer Research UK last year. And we kind of were about to, well, we did launch in March, but then the world changed, and it seems like everything has kind of slowed down a little bit, but we're working away in the background to get things off the ground. So I'm going to give a little bit of an introduction. You guys don't really need this background, but I think the points are worth making that, meso in the UK is the lesser known epidemic, if you like. And for decades it has been underfunded. We've really suffered relative to our other well-deserving cancers in terms of pounds spent on lives lost. And Dean's talk is kind of inspiring because we start to see things actually changing. We start to see maybe 1 year survival starting to shift, but it remains a cancer of unmet need, and there is a kind of global epidemic on the horizon, because the images you see there are young children and adults working in the BRIC countries, so that's Brazil, Russia, India, China, where there is really no asbestos control, and so there is a kind of an epidemic looming. Dean's kind of talked us through the kind of exciting developments over the last 12 months, but really, this is a review article published a few years ago that just looked at the numbers of trials active and what phase they were at, and there's a positive trend there, which is a good thing. You've got more trials running, but you can see in terms of where those trials are that very few studies or very few research questions or drugs get to phase 3. And phase 3 is, of course, where we test whether a drug actually works better than what we currently do. And there are lots of reasons for that. But one of the reasons, I think we would all agree, is that we have not, over the last 20 years, understood the disease in a way that's allowed us to really design therapies in the best possible way. And therefore, we're more likely to fail in patients. It's much better for us to fail in the lab before we go into a patient. Our understanding of the disease has improved incredibly, and you'll recognise some of the broad groups of drugs here. Immune checkpoint blockers and arginine deprivation were picked up by Dean. Viral therapies, probably not going to excite anyone at the moment, but oncolytic viruses are another strategy that we've tested. And anti-angiogenesis drugs have certainly been a kind of significant flavour over the last few years, and antigen-based vaccines also. The difficulty has been translating our knowledge from the laboratory, or our knowledge about a mesothelioma tumour cell, into designing an effective drug. And so for those of you who are interested, there are these kind of important publications that have really allowed us to really describe the mesothelioma genome quite comprehensively now. And the figure that's plotted there in simple terms is the number of mutations per cancer cell by different cancers. And mesothelioma is the third one along on the left. So it's a cancer that doesn't actually have, relatively speaking, as many mutations as others. And in many ways, that's a good thing. And we would think that if we could understand that mutation, we could drug that mutation and we could cure the patient. The real challenge in meso is that the mutations that occur are not mutations in oncogenes, and those are genes that drive a cancer's growth and life cycle. They are quite commonly mutations in what are called tumour suppressors. And tumour suppressors are a bit like the brake of your car. Not having a brake doesn't necessarily mean your car's going to accelerate. And so it's been difficult to understand how just not having the brake applied, not having a functional tumour suppressor, has translated into a cancer that kills patients so quickly. And it's therefore been quite difficult to design therapies. And we've extrapolated a lot from other cancers rather than had bespoke programs like Dean's. And the genome is also dominated by a very obvious inflammatory signature. Inflammation in the immune system seems important. So we really designed Predict Meso really from that position. And, you know, this scheme at the top that you'll recognize, it's a little smaller on my screen, but you might, I suspect you can get the idea, where we have usually 30, 40, thank you, 50 years of inflammation. And then at some point, we have the evolution of mesothelioma. And it's not clear what happens there. There is clearly a final critical last mile where maybe the immune system fails to stop killing those little tiny mutations that we all develop in our life, or some other inflammatory process develops, or something happens. And understanding that last mile gives us an opportunity to drug patients more effectively, but also perhaps to intervene earlier. And in the second little figure there, you've got a description of something called benign asbestos pleural effusion. So that is a phenomenon that the chest physicians here will recognise, where patients present with fluid and it looks like it could be meso. We biopsy them, but it's not meso, it's just inflammatory change or fibrosis. But we all follow those patients up because we know at some point they may develop meso. And there are some subtleties to that as to whether that was really a meso all along, but the reality is that that window of opportunity is that one that we can exploit, because we often have inflammatory biopsies in these patients just before they develop a meso. And that paired material is potentially incredibly powerful for us understanding what changes. And the other really exciting thing that's happened in the last 5 years, 3-4 years, is Marion McFarlane's lab in Leicester have a mouse model, which is kind of described by the little cartoon at the bottom. And in their mesothelioma mouse, if they inject asbestos into the pleural space, they can watch over time and they can actually demonstrate that some of the genetic features that we see in the clinic in established mesothelioma tumours are actually present in the pleural space before the cancer invades, or even before it becomes cancer. And so there is potentially this opportunity to intervene early, because some of these events are maybe obvious in patients before we see them. So the accelerator was funded last year and provides us with £5 million over 5 years from predominantly CRUK, but also CRUK's compatriots in Spain and Italy. And the aim and questions are reasonably simple in that our primary aim is actually to build these critical tissue cohorts of benign cases matched to mesotumor, and also to build technologies and infrastructure. And the infrastructure particularly includes really robust, well-designed preclinical models, so that we at the end of 5 years can get to a point where we have more drugs ready to go into a mesothelioma trial network, a bit like Dean's MIST platform. And we would really see these things as being running in series and together. And the questions that underpin that really are: how does inflammation evolve into meso? And what are the key molecular events? And importantly, what are the vulnerabilities associated with those events? Dean gave the really great example of arginine deprivation, which is a vulnerability in ASS1 mutated mesocells. And those are the sorts of things that we would be looking to see. And we also want to try and understand who might the best patients be to recruit to even chemoprophylaxis studies of high-risk asbestos-exposed patients. And so we need to understand risk, and we need to understand response, and I'll kind of talk you through that. So this is our map essentially of the network, and given the nature of the funders, we are predominantly based in the UK, Italy, and Spain, although we have collaborators in Belgium, and we have a significant collaboration in Brazil, in imaging, and that's important because Brazil, of course, are one of the big countries that are going to be faced with meso in the next few decades. And we've already had contact from groups in the US and other countries, and it is a network that we hope to grow and expand and share. And if you just look at the image in the center there, the network is a network in the sense that we aim to share everything, and that includes data and tissues and models and information. And it's also a network of work packages which are linked together. And so we have 5 work packages. And so in the time I've got, I'm just going to show you a single slide giving you a kind of brief overview of what each work package is going to try and do. So the first work package is the really critical step in actually building these cohorts of benign meso matched tissue pairs, and using that tissue to understand downstream in the other packages how that meso has evolved, and also to build models using that tissue. There's a really important data sharing piece. Crispin Miller in Glasgow, who's our bioinformatics lead, is essentially building a sharing infrastructure, and we'll use ICGC Argo to share a lot of the sequencing and other data out with the network. We've placed a lot of importance on developing other new researchers. So we have a series of schools and seminars planned, and we have 5 PhD students who are spread across the countries, including 2 that are coming from Brazil. And the reason for that is it's for the sustainability of this and for this to grow. It's the people that will make it grow, much like our clinical network in Scotland. And so us developing these people, I think, will help us move the thing beyond the life of the actual accelerator award. The schematic there is just illustrates that the tissue collection effort is necessarily complex, in that we are collecting prospectively, and we'll do a large prospective study in the UK involving 25 centres, where we recruit patients with benign pleurisy and follow them up. And we also will have a retrospective element, which is going to be really housed within Mesobank, which is Robert Rintoul's labour of love. And Robert is going to build those tissue pairs for us in Cambridge. So the second work package is really our omics. And so the intent here is to use some really incredible researchers across Europe: Andrew Biancan in Glasgow, Susana Cedrez in Barcelona, Manela Steller also in Barcelona, and Seth Kofelt in Glasgow, to help us understand what processes are allowing or driving the evolution of meso. And so those broad groups are the genome. So those are really the development of mutations in the DNA, the epigenome, which is more to do with the switching on and off of genes, and the transcriptome, which describes what proteins are expressed and whether those are actually active in the cell, and then the immunome, which is really describing the immune landscape around a mesothelioma tumour. And that's really critical in meso, because our pathologists constantly tell us that most of the biopsies that we send them are stroma, they are microenvironment. It's inflammatory cells, fibroblasts, and the odd tumour cell. And so we hope to try and understand that over the 5 years. So work packages 3 and 4 are together on this slide, and this is a hugely shameful simplification, but essentially the material that we generate from the patients will be used to generate preclinical models. And preclinical models essentially have various forms, but they can be as simple is the wrong word, but they can be as clean or as pure as a patient-derived cell line, where it's just tumour cells. That's harder than it sounds. or it can be an organoid or an organotypic model where we try and have a three-dimensional model that has tumour cells but also has some of these important immune cells. And then we have more integrated models that might be within a mouse, and particularly within a mouse with a functioning immune system, which again is quite complex and difficult to achieve. The thing that will help us is that Daniel Murphy, who's on this slide, has over the last 10 years developed, you wouldn't like me to say perfected, a genetically engineered mouse model that has got an intact immune system that develops mesothelioma in a way that we would recognise compared to the human disease. And it will really function in work package 4 as the test bed for new drug target combinations that emerge, that we can actually test them in that mouse before they go into patients. And then the final work package is split into 2 parts, and I mentioned risk at the start. So the risk element is the top little cartoon there with the orange boxes. And so it will be tied to the meso-origin, to the prospective tissue collection programme. And what we will be doing there is in all the patients who have benign pleurisy who we recruit, or a proportion depending on the biomarker, we'll collect biomarkers in blood and in exhale breath and in perfusion MRI, and then follow the patients up. And we hope to be able to conclude with a signature that actually identifies the really highest risk groups that you might use, for example, as eligibility criteria for a trial of a new drug that emerges from the back end of the pipeline. And then the last bit is the little cartoon at the bottom with the pink boxes, is we have a whole work package focused around artificial intelligence, and particularly its use in imaging. The images, could we just blow that slide up just a little bit, just to show so still not huge, but hopefully you can see the panel at the bottom shows essentially CT scans with green rings, and the rings are either axial or coronal views of mesothelioma, where the colours show the tumour that has been outlined. And that process is incredibly laborious for a human to do, but we've, over the last 2 or 3 years, worked with Canon to develop an artificial intelligence algorithm that we've trained to do that, so the green images you see there are actually have been drawn by an AI system that has never seen that tumour before. That you can't see it, but the image to the left is actually in red, is the human drawing that was used to train it. And so we're gonna, within Work Package 5, we'll pull together 1,000 CT scans from across the UK using the NCME network. So we're leveraging all their infrastructure. NCME is the National Centre for Intelligent Medical Imaging, and Fergus Gleeson runs that. And so that will allow us to hopefully generate an AI tool that could be used not just, it really is for clinical deployment or for making clinical trials cheaper, because you don't need a radiologist to report your response. Oh, there you go, fancy. Yeah, there you go. So that was it really. I'm going to kind of wrap up. I mean, some of these comments are not just about the accelerator, but are about, the clinical network. I think we have made a lot of progress. We've heard some challenging things, but we've also heard some really positive things. I think Dean's talk is incredibly exciting with what's on the horizon next year. And a big thank you to all those involved, including our audit staff, our QPI staff, our admin, and all the clinicians. The challenge really for Predict Meso and for research, I think more broadly, is better preclinical science and getting better drugs into trials. The title of Paul Krause's book is really what we're trying to get to, which is surviving mesothelioma. And I think This year, I think we're probably a little bit closer than we were last year for various reasons, even though the pandemic has been a bit miserable. And I'm going to stop there, and I'm happy to take any questions with Dean.
Kevin, thanks very much for an excellent talk. I think it's a really exciting project, really ambitious, and I'm impressed that you've even got it together and got funded. So congratulations on that. But I'm even more excited to hear the results in 5 years' time. So we'll book you in, I'm sure, for a repeat visit then, and you can update us on the results. We've had some excellent questions coming through on the chat. So if it's all right, I'm going to try and moderate some of those if people are happy to stay with us a bit longer. So Dean, a few really good questions for you, if that's all right, particularly around the chemotherapy. So from Sarah Morgan, what's your opinion of nivolumab now being available on the NHS for second-line treatment? And should we be doing PD-L1 more routinely in meso in the same way we do for lung cancer?
So I think the answer to the first question really is it's great. Having nivolumab as a less toxic second-line therapy is a good thing. There's no licensed treatment, so giving vinorelbine is off-label. If you give nivolumab, it's still off-label, but at least it's a licensed drug somewhere in the world, and that's Japan, and it's very well tolerated, of course. So the big question is, that's going to be something made available until March of next year by the NHS. Based on whatever we see in Confirm, we'll need to know whether or not NHS will be willing to continue to offer that if the trial reads out favourably. In terms of PD-L1, at the moment, the evidence suggests actually that it probably isn't a useful predictive biomarker. I think the big difference with lung cancer is that it is clearly a very good predictive biomarker. If you look at something like Study 24, which was it actually led to frontline standard of care with pembrolizumab in high PD-L1, about 50% tumor proportion. We are not seeing anything like that yet in mesothelioma, where high PD-L1 selects for benefit. Confirm will be interesting because that placebo control arm is slightly different. We don't know what PD-L1 does in chemotherapy. It makes it work less well. But with the placebo control, we can get a clear idea of what the predictive and prognostic interactions are. And so that's something we will definitely present data on that in all 300 odd patients within that trial.
Great, thank you. And I think Kevin had a question for you, but I think you probably answered it during your talk, or at least the end is, what's your prediction on EMA and NICE and SMA approvals for first-line ipi/nivo for non-epithelioid? And I know from a clinical perspective, we're hoping it's going to be before August next year that we get access to this, but just wondering what your thoughts are on that.
Yeah, so I mean, the FDA had their first crack at the whip and they gave it across the board. As we know, the US is very different from many parts of the world where health economics plays a really important role. And particularly where in that subgroup setting, we're not seeing a difference between the epithelioid for the immunotherapy versus chemotherapy. It'll be interesting to see what NICE says. I would imagine actually that the non-epithelioid is a shoo-in, that's going to happen. It's got to happen based on the hazard ratio, but for epithelioid, it's still up in the air really as to whether they'll go with it. I thought the FDA would give it. I think the EMA may actually give it. I think there's a better chance perhaps than NICE, but who knows? I'd like to see patients having access to it, to be fair, either way. I mean, there's some patients with poor renal function, for example, who really should not be getting chemotherapy or can't.
Yeah, excellent, thank you. A question from Malcolm. Will, it looks like it's better to be diagnosed with a non-epithelioid disease. Maybe these patients could be considered for trials of surgery with either neoadjuvant or adjuvant chemo and immunotherapy. I don't know your thoughts on that.
Is that to myself or?
To you, Dean, yeah, sorry.
Yeah, so non-epithelioid in surgery is a controversial area, I think. Very invasive tumour, very proliferative, much shorter time to progression following surgery. It is true that if you give the immunotherapy, you might expect a longer time to progression. But, the question then is around the ethics, I think, of having a radical surgical procedure being rescued by immunotherapy, if that makes sense.
Yeah.
So, it's a difficult problem. I mean, reducing the volume of the tumour and essentially creating a lower volume of residual disease might have benefits in itself. And so it may be a study that we see coming in the near future.
Excellent. And then I think we touched on this earlier in the afternoon, actually. So a question from Sarah Morgan, who's one of our colleagues in Wales. You've got huge experience, Dean, in delivering clinical trials for mesothelioma, and obviously Kevin, you as well. But much more from an oncology perspective. How can we reduce the barriers for our patients to get into clinical trials? So this is something that I know Kevin gets probably really bored of me saying. So I now live up in the far north of Scotland, having relocated from London, where it's very easy to get people engaged in clinical trials. And it's a constant frustration for us. And I'm sure for a lot of colleagues who live with more or who serve more remote and rural populations, it's not about the cost. It's not about the funding the travel. It's actually the burden that takes on the patients and their families if they've got a life-limiting malignancy. The commitment that goes into a clinical trial with additional travel is really quite huge. So what do you think we should be doing, both from a clinical perspective, from the standpoint of the people who design the clinical trials and able to deliver it, as well as what we can do to support patients to engage with the trials that are on offer?
So I think the answer to that question is we need more phase 3s. When you look at the sample size around the phase 2, you might be looking at 20, 30, 50 patients. And trying to deliver that through 30 centres doesn't make sense economically. With something like Confirm or Vim as an example, we're looking at 25, 30 centres. I still don't think that's enough. I mean, as you say, and actually I should mention this, that Mesothelioma in the UK played a really important role actually in providing funding for some patients to travel actually into that phase 3. We tried with Confirm to involve all 4 parts of the United Kingdom. So we're very pleased to say that Belfast was involved. We were able to get Northern Ireland on the map. Of course, Scotland were key actually to the study. But that's the only way. We need phase 3s. And that's the challenge, because of course you've got to have drugs that are active at phase 2 to get to the phase 3. So it means putting these drugs through these early phase trials, finding signals quickly that look as if they're going to be meaningful, and then rolling them out. I suppose the second thing is actually related to that, but it's attracting industry. They can do things pretty fast. And we see in lung cancer that actually the UK is not on the map very often when it comes to the big, high impact international studies. Not always. Some studies, yes, but not as many as I'd like to see. And I think if we can get industry to be involved and work with us, then we'll have those rapid commercial, geographically widespread studies available to all patients, hopefully.
I think one of the things I learned in COVID, and perhaps one of the positives that have come out of it, is really the speed with which a lot of clinical trials came up and running. Recovery, I think, is a phenomenal success story for clinical trials. The number of sites that were open and the speed with which that came on board, and it was able to react to results. And I realise that's a pandemic, and you're not going to be doing that with non-emergent conditions. But I think, from a trials perspective, I think we need to really think about how we deliver them more effectively for our patients. So I'm in full support of that as to what we can do moving forward. Don't worry, Kevin, I do have some questions for you.
No, I was actually going to just jump in, because I think recovery is a really good example, because the thing that made, it's a pandemic, but the thing that made that possible, or one of the things that made it possible, is that you're using repurposed drugs largely. You're not using experimental medicines. And so we have woven that from the start into Predict, and that the drug screening that we would be doing on cell lines to candidate drug targets would be, primarily trying to use, if possible, FDA approved EMA. There is a list of many licensed therapies. And so if you can demonstrate activity from an antibiotic or hydroxychloroquine, God forbid, dexamethasone, you can deliver that trial at scale in any centre. You don't need a trials unit to deliver a very complex immunotherapy agent. So I think that analogy is a good one.
A question, Dean, for you. What routine sequencing do you think teams should now be doing and capturing irrespective of trial portfolio?
Yeah, it's a difficult one, that, actually, because we're doing sequencing within the trial context. Outside of trial, particularly those patients who have some form of legal compensation, I think it is helpful to get a kind of a profile of the molecular landscape. As papers like the MISS papers come out, with PARP, for example, there won't be any difference between vinorelbine, nivolumab, as from the earlier studies, and rucaparib. Actually, these are drugs that will have had a phase 2 trial attached to them with data that you may consider positive or negative. And the question then becomes, do you try to identify a biomarker to say that drug is the likely drug for you to have? So we are seeing more BAP1 and P16, CDKN2A screening in standard molecular diagnostics. We're doing that now routinely. I think we're quite late to that game, actually. Andrew Nicholson in London has been doing that for quite a long time. But that alone, given that these are the big, low hanging fruit, BAP1, CDKN2A, these things may actually end up emerging in the diagnostic panel, allowing us to not have to think about sequencing. But I'll come back to, again, McMay, actually. The patch1 is a rare mutation, but it's an actionable mutation. We do see BRCA mutations, as we know, are clear indicators for sensitivity to PARP inhibitors. And I think that when an individual has the resource, or a centre has a project or something like that, if we can screen, like in Memorial Sloan Kettering in the US, what we'll have then is that opportunity to see these potential targets, and maybe have more McMays, actually, who we can offer bespoke treatments to. But I wouldn't advocate systematic NHS, NGS screening just yet. We don't know enough, I think, to justify that.
Excellent. So I think that's, unless there's any other questions coming through on the chat, I think that's covered. Unless I've missed any, please do put it in the chat box if I've missed you by mistake. But I think that's covered the questions on the chat. I know, Dean, they were mostly for you, but Kevin, I did have a couple of questions, if I may, very briefly before we finish off, and I hand back to you around the accelerator program. I think this is hugely interesting, as I've said before, but I'm particularly interested around the progression, or the potential progression, of the benign pleural disease into mesothelioma. We all know as clinicians it's there. Our clinics are not, we've all got our fair few patients who've presented in this way. How realistic is it, do you think, that you'll be able to predict when such a small percentage of patients progress to mesothelioma? How realistic do you think that's going to be? Are there any early signs of any early ideas as to what might be the triggers?
You mean the actual underlying science that affects that evolution? I think we don't know, because we don't have, in humans, we haven't seen those paired samples analysed at any scale. Marion McFarlane's lab that I kind of illustrated, they had shown hypermethylation of CDKN2A, which is the event in their model that then precedes the then evolution into meso. Whether that's driving or whether that's a passenger is kind of uncertain. I think there's it will take probably, we're going to recruit maybe 600 patients with benign pleurisy in the UK and follow those over 5 years, and we're only predicting an evolution in about 60%. And in reality, I think a large number of those evolutions are not evolutions at all. They're people who had a first biopsy that was negative, that had inflammatory tissue in it, and then a few months later they have a biopsy that shows cancer. So it's a slightly different nuance, but essentially it's still anatomically different and phenotypically different tissues that show a change. So I think it's a long answer to a question, to an answer. It's a long response, but I think the answer is we don't know what those events are. I think the theory is that it's a failure of immune surveillance in some form. But
great, thank you very much. Well, I don't think anything else has come through on the chat whilst we've been talking. So, oh no. I think one final message, which is really a message of support. So thanks, Kevin, for your talk. Amazing you were able to get the Predict Meso network study into Scotland. Sounds like an excellent project to involve Wales in as well. So I think if there's no other questions, that just leaves for me to thank Dean and Kevin again for really excellent talks, really informative and I think very exciting. And I'm going to hand back to Kevin, who I think is just going to tie up the meeting. And again, a big thanks to everyone who stayed on and listened, and apologies we've overrun slightly.
Thanks, Beth. Yeah, I hope people enjoyed that. The thing with these webinars, it's not like a conference where you can look out in the crowd and you can tell if anyone's still there. I mean, we could just be talking to ourselves. But I hope there are some people still there. I mean, I've really enjoyed today. I think there's lots to be excited about, not least the fact that we had over 100 delegates signed up, and meso is on the agenda, and it's something that is at the front of people's minds, and I think there's an infrastructure to support the patients and a way forward. And I think we've got a really fantastic network of researchers that can actually move forward some of these big questions, and I think you are seeing that in what Dean's presented. I think, you know, things are on the up, and I think we can look forward with a bit of hope as long as we get rid of the dreaded COVID. So just, I guess I do want to thank Elaine Smith and Anna Morton, who are our MDT coordinator and project manager, who've done a lot of the work in pulling this together. And I want to thank our sponsors for the meeting. So Thompson's and Digby Brown and Lockett and our AV colleagues. They've been fantastic. And I think I will just draw it to a close. Obviously, thanks go to Macmillan, Meso UK and Clydeside Action, who've funded the actual network. But I think we'll hopefully see you next year, and perhaps we can do it face to face. So goodbye.